Thank you very much for the opportunity to present my work here today. As many of you would know, HLA-C602 is one of the main risk factors for developing psoriasis. And it is an MHC class I molecule which presents peptide antigens to CD8 T cells and NK cells. And we specialized in studying exactly those peptides using an immunopeptidomics workflow and mass spectrometry. This allows us to identify the range of peptides. And using this technique, we were the first to show that HLA-C6 presents predominantly 9 amino acid long peptides with arginines on anchor positions 2 and 7 and hydrophobic amino acids on the C-terminal position. With our collaborators, we were also able to show how the model psoriasis antigen ADAM-TSL5 binds in the groove of HLA-C6, and the ternary structure with the TCR is currently under revision. The main question that remains to date is what unites those patients that are HLA-C6 positive? What is the common antigens that trigger immune responses? To answer this question, we first did immunopeptidomics on melanocyte and keratinocyte cell lines, and we found that, again, we have this 9 amino acid long predominance and also the typical HLA-C6 motif. But in contrast to previous work on B cells, now we have over 30,000 peptides derived from skin cells, and that is reflected by a high number of peptides that are derived from proteins found in the skin and also from psoriasis-associated proteins. That's also reflected in the pathways that we find, such as melanosome formation, epithelial differentiation, cell adhesion, and so forth. But then we went one step further and also established a HLA-C6 transgenic mouse model to study this. And the mice that were used for experiments were heterozygous. That means they had a human HLA-C6 molecule, but also still the mouse MHC class I. And due to the lack of time, you're going to have to take my word for it that when we phenotyped for all the different components of the antigen processing and presentation pathway, whether they were human or mouse, they were expressed as we would expect. And that then allowed us to use the well-known Imiquimod mouse model of psoriasis. And then on day 7, when psoriasiform skin was fully established, we were able to collect lesional, non-lesional skin, but also the unrelated spleen to do, on the one hand, immunopeptidomics, but also to do flow cytometry. And in flow cytometry, we found that the mice express, the splenocytes from the mice express HLA-C6, and although you might think this is a low level of expression, it is physiologically low also in human cells because HLA-C molecules interact with NK cells and have to be kept in check. But our mice actually express very high levels of HLA-C602 intracellularly, and it is also physiologically upregulated. And when we then analyze the organs from those mice with immunopeptidomics, we found over 4,000 peptides from those mice. And importantly, they again featured predominantly 9-mers and again the very typical HLA-C6 motif. Most importantly, the predominant 9-mers, 85% of them matched either completely identically or with one amino acid difference to one of those previously found over 30,000 peptides that we found from human cells, which bodes well for any translation. And what was very interesting for us is that over 300 peptides were found exclusively in HLA-C6 transgenic skin, not in the spleen of those mice, and also not in wild-type mice. So we are now able to narrow down the candidates and to look at them further. And in the next step, what we are currently doing is we're validating them against T cells from patients. So what we could do is take skin tissue resident memory T cells, but also peripheral cutaneous lymphocyte antigen positive CD8 T cells, and then analyze them in single cell TCR sequencing. And what we find is that in the lesional epidermis, we have oligoclonal expansion, which is a sign of antigen-driven expansion and antigen-specific expansion. And this is very particular to lesional epidermis because neither in lesional dermis, nor in non-lesional skin, nor in PBMC do we see those clones. And this is where we're at now, and we hope that we can find new ways of targeting those TRMs via an antigen-specific way to get closer to the cure. And that just leaves me to thank all of our collaborators, the students who did a lot of the job, our clinical collaborators, and the funders. Thank you very much.
Any questions from the audience? I mean, you were looking at the clones. Did you check in previously, given the talks that we heard before, in previously lesional, non-lesional skin, if you find the clones?
No, that we haven't collected yet, but that's a good idea. We would expect that there would be fewer, but probably one or two still sticking around from all that we know from literature. Yeah.
At least I hope after the talks this morning. So if not, then we'll move on for the sake of time. Thanks very much.